Hi, my name is Isabelle Dingli.
Hi, my name is Andrew Naugle, and today we'll be talking about Sudarsanam et al. 2020 paper for Mike Orrell's group's presentation.
The paper is entitled Cofilin Regulates Axon Growth and Branching of Drosophila Gamma-Neurons. To understand the cofilin regulation of axon growth and branching in gamma neurons of Drosophila, it is important to understand the basics of the neuron cytoskeleton structure and the neural growth cone which guides axonal growth in both regrowth and the development of neurons. Axon growth and branching is largely dependent on the actin cytoskeleton. The actin filaments are composed of F-actin, which is made up of monomeric globular G-actin that binds together in a specific direction to create a polarized strand. The barbed end of the filament is positive, whereas the pointed end of the filament is negatively charged and is associated with ADP. G-actin can bind to both ends, but preferably binds to the positive end, while the negative end deactivates and is depolymerized. When this reaches the right balance, it is called filament treadmilling. For the actin to break apart in the treadmilling, cofilin is the main protein promoting depolymerization. However, the actin can depolymerize without the help of cofilin. In Drosophila, the cofilin is named twin star. It is a severing protein which breaks apart F-actin into G-actin. Twin star binds to the ADP end and increases the rate of depolymerization. The local depolymerization of actin causes the cone to turn away from that area. Within the cell, in between the actin cytoskeleton filament, lies the microtubule cytoskeleton, made up of tubulin. For the axon to continue growing, the microtubule has to have room to grow, hence the need for the actin cytoskeleton to be rearranged.
Thank you, Isabelle, for the background context on the structure and function of neurons and neuronal remodeling. Before introducing the research study, I will be defining a few crucial terms. Isabelle mentioned the growth of gamma neurons, but it is important to note that gamma neurons specifically innervate the muscle spindles. These gamma neurons compose the mushroom body, which are parts of the Drosophila brain essential for olfactory learning and memory. The mushroom body, or MB, were used as a model to study developmental axon regrowth in this paper, specifically to explore young developmental neurons in Drosophila. Now regarding the questions Sudarsanam et al. Asked and attempted to answer throughout the paper, there are a few common themes to note. Firstly, it is made apparent that in the field of developmental biology, there are several concepts that are not really understood. An example of this highlighted in the paper are the specific mechanisms that control intrinsic axon growth potential and regrowth. Therefore, this paper seeks to look at these mechanisms by synthesizing past paper knowledge with its discovery of how twin star, or TSR, promotes axon elongation as well as branching, specifically during the developmental remodeling of gamma neurons. However, the paper was not clearly able to conclude whether these two functions were linked or if they utilized the same mechanisms. Overall, this paper sought to answer how the role of Cofilin, specifically twin star's molecular machinery, controls axon regrowth, as this is not really known.
Regarding the main findings and take-home messages from this paper, it was noted that TSR mutant axons demonstrate growth defects both in vivo and in vitro, and also exhibit actin-rich filopodial-like structures at the failed branch points in vivo. The process of regrowth following injury is separate from remodeling, but it shares similar mechanisms, such as the branching process. We have known that cofilin plays a role in depolymerization and hence in the remodeling process. However, this paper shows that in Drosophila, the presence of TSR does not affect the availability of G-actin, which is evidence against the idea of treadmilling, but that the lack of TSR does affect the ability of the actin cytoskeleton to branch and grow properly in developing gamma neurons. Here we learn that TSR is crucial for growth and branching during developmental remodeling of gamma neurons. The paper hypothesizes that instead of TSR's primary role being to assist G-actin availability, that its primary role is in clearing away the F-actin to allow protrusion of the microtubule cytoskeleton, suggesting further studies look into this.
Thank you, Isabelle, for highlighting that this paper developed an agreement of Bracke's study in 1999 by hypothesizing that Cofilin promotes axon extension by clearing actin to allow microtubules protrusion. With this hypothesis, the paper was able to elaborate on axon formation with respect to twin stars' role in axon regrowth of Drosophila mushroom body γ-neurons. Regarding the major findings within this paper, the role of twin star during axon regrowth and branching were met. Sudarsanam et al. Discovered 5 findings revolving around the role of twin star during axon formation. First, they found that twin star is required for axon growth of mushroom body γ-neurons. They also found that twin star promotes branching of adult γ-neurons, as well as how twin star mutant γ-neurons show reduced neurite dynamics. With this, they also found that twin star does not promote regrowth by increasing G-actin, as mentioned earlier by Isabelle and I. Moreover, they found that microtubule localization is disrupted in TSR mutants. Out of these 5 discoveries, the most important one to highlight, separate from the overall conclusion, is that phosphorregulation of twin star is critical for axon regrowth and branching. This can be seen in the following figures as the expression of the non-phosphorylatable TSR S3A, which was presumably constitutively active, resulted in partial growth rescue. Therefore, it seems that TSR is required for regrowth independently of its requirement during initial axon outgrowth.
Thank you, Anda, for the description and summaries of the results. On to why these findings are significant. Though in Drosophila, these findings have strong implications for gamma neurons in humans and medicine that could be used and made to treat mutations in neuronal growth. Neuronal remodeling is a conserved mechanism, and as such, the discoveries made in Drosophila set a precedent for future studies in humans. Furthermore, Cofilin has previously been thought to only have a role in the depolarization as a severing protein, whereas this paper has shown that its role in axon growth and branching is more significant than its role in creating G-actin availability, changing the way we have previously thought about cellular movement and growth. There were many different variables being tested throughout these experiments. To test each, the researchers utilized clones and isolated each variable being tested to its own experiment. One of the experiments compared TSR null homozygous mutant clones to wild-type Drosophila clones expressing TSR. Using clones and mosaic analysis with a repressible marker ensures that any other factors in vivo would stay the same. Comparing these to third instar larvae to see if the discrepancies were due to an issue in regrowth or outgrowth. In other phases of the experiment, single-cell clones were used instead of neuroblast clones, overall providing the experiment with neurons in a variety of different life stages with different levels of outgrowth, regrowth, and different intracellular factors due to temporal reasons, all while keeping those outside factors the same between the tested populations. Due to the variety in the experiments, they were also able to show which mutants were able to rescue the growth defects of null mutants and which have partial rescue effects. The paper did address the potential issues with heat shock protein or with the GAL4 drivers that could have caused discrepancies seen through the ages of the Drosophila, as well as issues with RNA and other molecules that were added. Throughout the paper, many different twin star mutations were studied in different types of clones, making the conclusion for the role of twin star overall a bit less solid, as each mutation had its own effect on the outgrowth and regrowth of axons and was studied individually of the other twin star strains, with some mutations being used in some experiments but not others. The conclusions and summaries drawn are overall implications of TSR, proving some prior thoughts wrong but not directly answering the initial questions posed in the research paper.
As it is known that science is always evolving, especially within the field of developmental biology, this paper was able to reference past studies which demonstrates the importance of the dynamic nature of collaboration within the scientific community. This paper did a great job with the rejection of previous ideas and served as a segue into future potential studies. Overall, this paper will serve as a reference for future research in the mechanisms that control axon regrowth within Drosophila and even maybe towards humans and other organisms.
Thank you for watching.
Thank you. We hope you enjoyed.